So I'll start. My name is Ana Maria, and I'm here to just give a brief introduction of who we are in LatinX, to talk a little bit about how this speaker series came about, and then give an introduction to the speaker that we're having today. That we were very excited about him joining us and telling us about his research. So as I mentioned, I am the co-chair of LatinX Abroad, and I'm the co-chair of LatinX Abroad. And I'm happy to be hosting the series today. We have been hosting the series now for a little bit over a year. And we are always very grateful for everyone that joins. Sorry, one second. So as I mentioned, this is an initiative that we had here at LatinX Abroad. As I mentioned, we did it over a year ago. And LatinX Abroad is an affinity group that aims to create these spaces of community building where we create allies and we work together as a community. Not only here have some pictures of some of the social events we do, but also to create those spaces where people feel comfortable talking and brainstorming ideas and just creating network and helping each other here in the field of STEM. As you know, Latinos in the field of STEM are a minority. So we like to create those spaces where we feel welcome and just be able to create great collaborations together. And as I say, our main goal is to empower our community. And that's where this idea of the LatinX speaker series came about. We noticed that there is a lack of representation of all the great work that we're doing in the field of STEM. And we all need role models that we can, follow and that we can feel comfortable reaching out and asking questions. So we came up with this speaker series where once a month we meet together. This is an open event for the public, so it's not only for people at the abroad, but we have people joining from different parts of the US, and actually we also have international people joining from different countries in Latin America. And what we want to do is create this space where research scientists from the Latin community can come here, talk about their work. We get to hear about it, and we can, reach out to them, ask questions, and maybe create this networking opportunity. Either you can communicate directly with us at LatinX, or you can always will reach out to communicate with our speakers. They are always open to hearing questions and receiving information from anyone, and just in general promote all the great things that we do in the community. So today we have a very special talk from Dr. Bravo. He will be talking about the neuroprotective and neurodamaging effects of the myognone cells and oxygen in different ocular pathologies. So we'll hear from him first, and then always feel free to reach out using the Q&A button. We'll be looking at that. And for those of you who are on the room at the Broad, we'll also have time for hearing questions from you. So without further ado, I will start introducing our speaker of today. So as I say, his name is Dr. Bravo Nuevo. He's an associate professor of neuroscience at the Philadelphia College of Osteopathic Medicine in Philadelphia. He has a PhD in neuroscience from the Australian National University in Canberra, Australia. He also did his postdoctorate at Harvard Medical School, and from there he became an assistant professor at a medical center in Wynwood, Pennsylvania. He now teaches neuroanatomy, ophthalmology, and molecular biology, and has recently created a medical Spanish course for the first and second year medical students. His laboratory also hosts about 25 students and 2 research technicians. His research focuses on ophthalmic diseases as retinitis pigmentosa, wet macular degeneration, retinopathy of premature glaucoma, and corneal transplant failure. So his team has recently been awarded a $2.3 million foundation grant that is helping him continue all the great research that he's doing. So we are very thankful to be able to be hearing about it today. So without anything else from my end, I give the floor to you, Dr. Nuevo, for hearing about your research.
Thank you, Ana Maria. Thank you to all of you for being here. It's a real pleasure. It's a great initiative. And I'm very, very, very happy and honored to be here and being part of it. All right, so look, that was my initial title, and then when I put together the presentation, I think if I talk about myanox cells and oxygen, it's just going to take 2 hours of your time. I'm not going to do that. So I'm going to talk about myanox cells, and maybe next year, if I get invited again, I can talk about the effect of oxygen, the good and bad effect of oxygen in some of the ocular pathologies. All right. So my talk will be about myanox cells mostly. First, I'm going to talk, this is an outline of my presentation. I'll be talking first about the lab. Then I'll give you a brief introduction to the eye and the retina. Then I'll talk about the myanox cells, some good side of myanox cells, the good aspect of it in some different pathologies, and some bad aspects of myanox cells, and then some work that we've been doing after all of that. So get around maybe 10 years of work here in this presentation, and a lot of effort from many students. As Ana Maria said, I am at the Philadelphia College of Osteopathic Medicine in Philadelphia. This is a multi-campus school. It's a postgraduate school. We have campuses in Philly, and we have 2 campuses in Georgia. It's mostly a medical school. It's an osteopathic school, but we also have a master's in biomedicine. We have some postgraduate degrees on psychology and a physician's assistant program. Now, the school has an interesting approach. For medical students, it's quite competitive to get into residency program. So research and publications are a very important part of getting into a residency program. So the school invests a lot of money into supporting research. And they do that. They encourage you to write grants, but they do have a strong internal funding program that helps a lot of the projects. Students are strongly encouraged to do research, and I have maybe the biggest lab for all PCOM campuses, and they are terrific. Many of them are doing this during their medical school, and every free time that they have, they spend in the lab. So some relevant things about our PCOM is where the first studies connecting infections beyond the olfactory nerve and dementia were done by Brian Bailin. This is relevant now with COVID on the long-term consequences that might have some of these infections through the olfactory nerve getting into the brain. Of course, the myanox cells were discovered here is one going to talk, and some famous alumni was Sean Connolly, was the clinician for Donald Trump, and Kenneth Ryland, who was the clinician for the White House for Richard Nixon. Okay. So my lab focuses mostly on the central nervous system. Most of our work is done in the eye and mostly in the retina, but we have some work in the cornea and the lens, and a little bit of work in the brain with around 20-25 students in the lab per year. Usually we have 2 research support technicians. They are shared between different labs. So sometimes we have 2, sometimes we have 1, but usually we get around 2. And we have established several animal models for diseases affecting the eye. Some we purchase, and some we have developed ourselves. We have models of retinitis pigmentosa, glaucoma, corneal transplant, wet macular degeneration, retinopathy of prematurity, epiretinal, pre-retinal membrane formation, and that has allowed us to collaborate with many biotechnology and pharmaceutical companies that usually we form partnerships with, because they're interested in doing some work in the eye, and they with time, because we cover so many diseases, they come to us. So just a brief introduction to the eye for those who are not familiar with it. The eye, the light comes through the cornea. It goes through the pupil, which is a hole between the iris. Then it goes through the lens. And in primates, and only in primates, most of the light focuses in the fovea. Rats and mice don't have macula or fovea. The fovea and the macula are within the retina. The retina is a layer of part of the central nervous system that is covering the back of your eye. And is where the light gets transformed in an electrical signal that will be sent to the brain to be processed. So the retina, just very briefly, if the light comes from the top in this picture, it has to go through a series of layers before it reaches the outer segments of the photoreceptors. Those are the rods and the cones that you probably remember from high school or some courses in college. Those are gonna capture the light, and those photons are gonna send a signal back to the brain. And that signal gets pre-processed in the nuclear layer and then sent through the ganglion cells, which have the axons are gonna form the optic nerve, and that optic nerve is gonna be sent to the brain and the optic chiasm, and for them to be processed. So you have three major nuclear layers in the retina, and these are important, so I'm going to be talking a lot about them. The outer nuclear layer is the one where the photoreceptors nuclei are located. It's right here. So cones and rods, outer nuclear layer. It's outer because it's the one that is the farthest from the center of the eye. The inner nuclear layer comes next. You have some glial cells, like Mueller cells there, and you have some pre-processing cells that have the nuclei there, such as bipolar cells, amacrine, and horizontal cells. Finally, that information gets sent to the ganglion cell layer, which, as I said earlier, the axons are going to form most of the optic nerve, and they are going to be sending that signal to the brain. Now, in my lab, because we do a lot of work in the eye and the retina, we need to do some specialized instruments that we can measure different parts of the outcome of our experiments. One of them is an optical coherence tomography. It's called OCT. This is what you can see here. And that measures the thickness of different layers of the retina. You can put the mouse, and it's a non-terminal method, so you can follow the same mouse through weeks. When animals get blind, in many cases, the retina starts to become thinner and thinner, so you can see how it gets thinning through the weeks. This is an image of an OCT scan. You might have had this done to you when you go to an optometry clinic or an ophthalmologist. You can see the different parts of the retina. This is the optic nerve. This dark, thick layer is the outer nuclear layer. That's where the photoreceptors are, and this is the total retina. And usually we give thickness values as the ratio between the ONL and the total retina. So assessing vision in mice can be difficult when you're talking about function, because of course you can't put a mouse in front of a chart with letters and numbers. They can't tell you the third row from the bottom. So we need to do some functional studies sometimes to see how what we see anatomically translates in how well these animals can see, or the effects that the way the retina process light, right? So for this, we use an electroretinogram. This is an ERG. You can see here, this is an anesthetized mouse. It's also a non-terminal disease. We put these mice overnight in a dark room because we want to, most of the cases, look at scotopic light. That's the response to bright light by the rods. And this machine is going to send a series of flashes, and we put some electrodes in the animal, and one of them is just laying across the cornea, and that is going to measure the electrical activity of the retina. And the way we measure it, it comes out, this is an electroretinogram. You get two waves. The first wave is, you can see here between the black and green arrow, that is the A wave. The amplitude of that wave usually reflects how the retina, and in particular the photoreceptors, react to light. So the larger that wave, the better reaction they have to light. The big wave is the wave that goes from the green to the red light, to the green arrow here. And that is the polarization of the inner retina, so mostly the inner nuclear layer and the ganglion cells. All right. A lot of my talk, or most of it, is going to be talking about myonox cells. Sometimes I will refer to them to G8, or you will see them in some of the figures, G8 and BA1 positive cells. This is because of an antibody we used to detect the myanoid cells. So when we label them, that's what we use. So anytime you see E8 plus or B1 plus, just remember we're talking about myanoid cells. It was discovered by Dr. Mindy George Weinstein and Jackie Gerhardt around 20 years ago. The name comes from the expression of MyoD, mRNA, and noggin protein. They also express BAY1, which is something that we can use. That's the brain angiogenesis inhibitor 1 is something we can use to specifically either deplete the myanoid cells or label them. They are found in almost every tissue in the body at low levels. They're important for the development of the embryo. Without them, the embryo does not develop properly. But once you're an adult, they're in low levels, but the numbers do increase when you have a stress or injury, and I will talk about that later. It's important to know that they can turn into myofibroblasts, and that is something that we're going to talk about that later as well. It's an important point to remember. All right. So what happens when you deplete an embryo from myanoid cells? This was initial studies done by Dr. George Weinstein and Jackie Gerhardt. They were embryologists. They are embryologists. They did a lot of embryonic work. And what they showed is there was a lot of defects in the embryo. But what is relevant for us today is that it led to severe malformations in the eye. You have microphthalmia, which is what you have here, when the eye doesn't fully develop. You have invaginations of the retina, as you can see here at the bottom right, and you have self-closure of the retina, you can see here. So it doesn't develop properly. Where are the myanoid cells located in the retina? Well, they are almost in every layer of or every tissue of the eye. You see them in the cornea, you see them in the lens. In the retina in particular, they're along the vascular beds of the retina, close to the nuclear layers. So as I said, Dr. George Weinstein and Jackie Gerhardt were discovering these cells, but most of the work was in a chick embryo. They really hadn't done much work on mammals. So we started collaborating around 15 years ago. And the first thing we wanted to look, it were these myanoid cells stress had, because we knew stress had effects on the myanoid cells in other tissues. We wanted to look what happened in a model of retinitis pigmentosa. Retinitis pigmentosa is a disease that affects mostly the rod photoreceptors in the eye. Those are the ones responsible for low light vision. When you go to a bathroom at night and things like that, or under the stars, it ends up expanding, and at the end the cones are going as well. And we have 60% of the causes for retinitis pigmentosa are due to genetic mutations. And there's murine models for retinitis pigmentosa. One of the most famous ones is the C3H mouse. And the C3H mouse is going to be blind by 6 or 7 weeks, and have all the photoreceptors dead, or most of them. And when we looked at this during different weeks, the number of myanoid cells that were in the retina in these mice compared to controls, the control background for these mice is the C57 strain. You can see here in black, so these are different ages for the mice. The number of myanoid cells in the retina in black is the ones that we found in C57 mouse, and in red is the ones that we saw in the mouse model of retinitis pigmentosa. So there was an increase of myanoid cells in the stress in the retina. When we look at the location of those myanoid cells within the retina, and up here you have, just as a reference, the picture that I showed you earlier. Remember, this thick layer over here is where the photoreceptors are. This is called the outer nuclear layer. Then you have the inner nuclear layer, and then you have the ganglion cells layer, which is here in the top, is the one that's going to have the axons to the forming the optic nerve. Remember, in retinitis pigmentosa, the layer that goes is this one over here. So the outer nuclear layer, that's where the rods are and the cones, and that's going to be gone. So, these animals are getting blind, so most of the stress is happening here. So when we look at the myanoid cell distribution in C57 control mice, we see that most of the myanox cells were around the inner nuclear layer, the inner retina. So either inner nuclear layer or some in the ganglion cell layer. So most around this area over here. And that's what you can see the chart pie on the left. This is the C57 mice, control mice, different ages. And you can see the distribution of the myanox cells mostly is in the gray and black parts of this pie chart. That is the inner retina, which is here. Okay. Now, when we look at the retinitis pigmentosa mice, these are the mice that, you know, this layer is going to be gone. We saw that most of the myanox cells were migrating to that area, as you can see here in red. That is the area in red that you can see on the right. So most of the myanox cells were going to the area of the outer nuclear layer around here. So they're migrating to the area of stress. We did several studies in different animal models that I don't have time to explain to you. And what we found is that not only the numbers of myanox cells correlate with the level of, with the location of the stress, but also the level. So if we ameliorate the stress, the number of myanox cells is going to go down accordingly to the stress level. Okay. So, you know, myanox cells either migrate or divide, but they populate the areas of stress within the retina. So the question that we wanted to answer next was, well, that's great. What happens if we remove them? So what happens if we remove myanox cells? For this, we wanted to use a model of retinopathy of prematurity, called in the mice the oxygen-induced retinopathy model. This is a model, is well established. I think it's Pat Damore in Harvard that first reported this experimental approach, and that's what, you know, VEGF therapies against wet macular degeneration and diabetic retinopathy were first studied. In this model, what you do is you keep the animals, so vasculature of the retina in mice, unlike in humans, develops after birth. So you don't need to induce a premature birth on these animals to look to have a mouse model of retinopathy of prematurity. So 7 days after they're born, you put them in a high oxygen concentration. And then that represses the signal, the angiogenic signal that forms some of the vessels in the retina. So you put those animals back into normal oxygen, there's going to be hypoxia, as the retina has the highest metabolic rate in the body. And that is going to lead to cell death, because there's not enough blood supply for the photoreceptors. And you know, also you're going to have neovascularization, which is not relevant for these studies, but is relevant for people studying wet macular degeneration and diabetic retinopathy. All right. You can intervene here, and that's what we did. So as soon as the animals open their eyes, what we did is we did a series of injections. And one of the injection that we did is we use an antibody specific against myanox cells. It's called the G8 antibody or the BA1 antibody, which specifically binds myanox cells, and we conjugate with complement, and that we showed in vitro that it leads to myanox cell depletion. So that's what we did, and what I'm going to show you is this is a cross section of the eye. On the left, you have the cornea, right here, and here this double layer that looks like a snake. Is going to be the retina. The thin layer over here is going to be the ganglion cell layer. The thicker layer over here is going to be the inner nuclear layer. And the brightest one, that one is the most outer layer, is going to be the outer nuclear layer. So you have the ganglion cell layer, the inner nuclear layer, and the outer nuclear layer. We labeled apoptotic cells, so cells that are dying. Remember, this is going to cause some stress in the retina. And those we use the tunnel technique, and that's going to be labeled in red. What you're going to have in green is the myanox cells. That's what we're going to be labeling. Anything else that is in blue is a nucleus. So what we're going to do is just do an overview of this control. This is a control that has been going through oxygen-induced retinopathy, and you can see some, you have some apoptosis labeled in red, and you have some myanox cells, and we can focus in a particular area, so you can see more detail how the apoptosis is labeled and how the myanox cells are, in this case, in the ganglion cell layer, in close proximity to some tunnel. And now, when we got rid of the myanox cells, remember the experiment here was putting the retina under stress and removing the myanox cells and see what effect it had in the retina. And again, you're going to have the myanox cells in green and the apoptotic cells in red. And yeah, so we do the same thing. This is the cornea, and now we start looking at the retina. This is the retina. Here you see a lot of apoptosis and very few, if any, myanox cells because we used a system to get rid of the myanox cells. So seems like successfully we got rid of the myanox cells. We can plot this in a chart. This is what you see here. On the left, you have cells per section. What you have in black here is the tunnel positive cells. Those are the cells that are dying in the retina. And what you have in white is the number of myanox cells. The red square is marking 2 bars, and those are the bars in which we got rid of the myanox cells. As you can see, you have very, very few myanox cells. But what it was striking for us is that we saw a lot of increase in apoptosis. So it looked like the killing the myanox cells led to an increase in apoptotic cells in a mouse model of retinopathy premature. But of course, the question was whether that cell death that we were seeing was just the myanox cells themselves, and whether it had an effect on the retina or was just a direct effect of the myanox cells being removed, leading to more cell death in the neuronal retina, right? So that was a question that we wanted to look at. So far, we know the myanox cells migrate to areas of stress within the retina, and we know that the depletion of myanox cells seems to lead to an increase in neuronal cell death. So just to confirm that, what we wanted to do is what if instead removing the myanox cells and killing them, we add some myanox. So we use exogenous myanox cells that we purify from another animal and add them into a different model of damage. And will whatever effect that have, will it be reflected in the retinal function? So will it translate into the function of the retina, or it's just dead and it has no effect on the function of the retina? So there's this set of experiments that we wanted to ask and see. For this, we use 3 models. The first one is the light damage in an albino rat. The second one is a model of glaucoma. And the third one, just because the retina is formed by neurons, but so is the brain, we wanted to look at brain and damage in the brain and whether what we saw in the retina translated into the brain. So we're going to look at the addition of myanox cells in each one of these models. So I'm going to explain them to you. The first that we did, I went to Sydney, where I did most of my PhD, to the laboratory of Jonathan Stone, and spent some time over there. And what Jonathan Stone had used is he used albino rats that have been raised under very dim light conditions. These were animals of 3 months, and they were albino. And the first thing that we did is before anything else, we did an ERG. An ERG, if you remember, was an electroretinogram, those 2 waves that we can measure function in the retina. And we wanted to do it before doing anything to those animals so we could get basal level of the ERG, so knew what to compare it to. Then we put the animals in 24 hours of high-intensity light. That is going to lead to a lot of blindness in these animals. It's going to deplete them from the photoreceptors. Okay. And then 4 hours after they received intravitreal, but that takes around a week for them to lose all the photoreceptors. So 4 hours later, after the intense light, we purified around 6,000 cells and we injected them in the eye, not in the brain. This is a mistake. We injected them in the eye. Of course, we had some sun controls, and then we did another ERG. All right, so intravitreal injections of the myanox cells and PBS, and we did another ERG to see whether there was an effect on the function. And this is what you can see here. So this is the superior part of the retina, and this is the inferior part of the retina. In a rat subjected to this light damage, most of the light is going to hit the superior part. As I say, rats are not primates, so there's no macular phobia in rats, but there is a hot spot. as you can see here. So what you can see here on left, A and D, is the superior and inferior side of the retina in a control animal. This animal did not suffer light damage. Here you have an animal that suffered light damage and was injected with PBS. And you can see the effect of the light, a heavy disruption of the photoreceptor layer, thinning, and disorganization in general. You can see barely see the outer plexiform layer separating those layers. And tunnel here is going to be labeling again cells that are dying. And then this over here is light damage with the addition of myeloid cells. Remember, G8 sometimes stands for myeloid cells. So because it's the antibody we use to detect them and purify them. So these were injected with myeloid cells intravitreally. And you can see there's less disruption caused by light. And the structure and histology of the retina and the outer nuclear layer stays as such. I told you earlier that we measure thickness in the retina, and that's what you can see here. This is the optic nerve, this is the superior side, this is the inferior side, and this is the thickness. So in black, you have the thickness of a normal rat retina that has not been subjected to light damage. Here in this gray line over here, you see the thickness of a retina of a rat that has been subjected to light damage, and you can see that it's thinner. And here is the effect that same light damage had when we injected myeloid cells. So you can see the hot spot in particular in the superior side is very protected. When we looked at the ERGs, the electroretinograms for this, you can see that the rats that never have been subjected to light have a very nice big amplitude response to the flashes, and they respond very well. Is what you can see here in black. When you cause light damage, you can see that response is diminished because they lost a lot of the cells in the retina. And when you inject myeloid cells in the vitreous, you can see that the response is protected. Okay, so that was for light damage. So in the light damage model, it looked at the myeloid cells were neuroprotective, and preventing them from the photoreceptors and the other neurons from dying. So next we looked at a different model. This is more of a physical model. This is a model of glaucoma. So glaucoma is a disease that is caused by the increase of intraocular pressure within the eye. So the posterior part of the eye is filled with what is called the vitreous, which is a gel-like substance that is there from the moment you are an embryo. But the anterior part of the eye is filled with what is called the aqueous humor. And the aqueous humor is constantly flowing from the back of the eye, behind the lens, all the way up to the front. And it's like an open shower. It drains through what is called the trabecular meshwork into the canalus phlegm. That drainage, just like in a shower, it can get clogged or things can obstruct it. And when you block it, because the aqueous humor is constantly being produced, intraocular pressure builds up. Yeah. And that intraocular pressure has only one way to exit, just like a balloon that you inflate. You know, the only place to exit is the valve. When you open the valve, all the air is going to go that way. So in the eye is the same. The only place it can exit is through the optic nerve. So pressure starts to build up, and that pushes the optic nerve back. Remember, the optic nerve is largely formed by the axons of the ganglion cells, as you can see here in red in this image in the bottom right. These are the axons of the ganglion cells that are extending and forming the optic nerve. So when pressure pulls, it pulls the axons, and it's going to lead to death within the ganglion cells, and what is called cupping, which is just making a push into an ulceration of that optic nerve, as you can see here on the right. So this ganglion cell layer is the one that's going to be affected the most. So the previous study, you saw how most of the layer was affected was where the photoreceptors was in the light damage in the albino rat. But here we're looking at a different layer, this layer over here, the ganglion cell, which is the one that has the axons that form that. So how do you generate a mouse model of glaucoma? So you just inject microspheres in the anterior cavity of the eye, and you're gonna block that trabecular meshwork. You're gonna block that kind of a slam, and you're gonna basically block the drain, and that's gonna build up. So we do that. We inject the beads, so they're magnetic beads, and then with a magnet, we pull them around and clog in all that trabecular meshwork. And by 3 days, you have high intraocular pressure. This is just a picture on the left depicting what the clogging of the beads does. And on the right, you can see an H&E staining of the trabecular meshwork with the beads clogging it. And you can see the obviously clogging this exit of the aqueous humor that comes from here. So that's how you increase intraocular pressure. And this is our experiment when we did that, and we had the right eye to be with glaucoma, the left eye without glaucoma, and we inject the myanoid cells, and we compared to PBS injection, and we did a series of controls. As you can see that after the beats, 3 days after, you have high intraocular pressure, and that high intraocular pressure will stay on pretty high for a few weeks, while the eye that didn't receive the beats is just gonna stay at low intraocular pressure, as you can see here on the bottom on those full lines versus the dotted lines. All right, so we kept them for 32 days. Now, for us at this point, it was because the animals do have myanoid cells, and we were injecting myanoid cells, exogenous myanoid cells. We wanted to differentiate between the myanoid cells that we injected and the myanoid cells that were there already, or endogenous myanoid cells. And for this, we designed a strategy with a double labeling. Once we purify the myanoid cells, the exogenous myanoid cells, we pre-label them in red with a dye that keeps the function. And then once we had the slides, the sections in the slides, we will go again with a specific antibody, BA1, and we label them in green. So what you end up having is the exogenous cells that were pre-labeled in red are going to be red and green, while the endogenous cells that the animal has itself, they're going to be only green. All right. And these are pictures of the same area using different filters. So this on the top, all these rows are just pictures of the same area, but with different filters for looking at different aspects. TM is a trabecular meshwork. Here you have the canal slam. Here you have the ciliary body where that aqueous humor is formed. Here you have the cornea. Here you're gonna have the ganglion cell layer right here. And here you're gonna have part of the iris, and you can see some of the beads here and there, that circular part right here. Just a reminder, the exogenous cells are going to be red and green, while the endogenous ones are going to be in green. And in these images, what I did is just the full triangle is going to be labeling the exogenous cells that you can see that they're red and green here in the channels, while the endogenous cells are just going to be with the open triangle here. And you can see some here, for instance, this green that is not here because it's just labeled with green, or down here at the bottom as well, here, here, and here. So those are the endogenous cells. So, we were able to locate those cells, and, when you have all the filters at once, they're gonna, if the exogenous cells are gonna have red and greens, they're gonna appear kind of orange yellow here, as you can see here in the merge image for them. All right, so what effect did it have, the addition of myanoid cells into this model of glaucoma? What you have here is the left eye. This is a non-glaucomatous eye, untreated. This is with an injection of PBS. This is an injection of myanoid cells. And on the right, you have the right eye, which we injected beads into. This is the outer nuclear layer. This is the inner nuclear layer. This is the nerve fiber layer, where on the ganglion cell layer is right there. As you can see in blue, which is H&E staining for the nuclei. And you can see the effect of glaucoma when the retina thins due to the loss of some of the axons and the ganglion cells. You can see how things compare to the left eye, but we did not see that thinning when we injected myanox cells intravitreally. We can look and count the number of ganglion cells, as glaucoma will lead to ganglion cell death. And you can see here you have the non-glaucomatous eye in black, the black bars, versus the glaucomatous eye, which are in white. As you can see here, those are the eyes that didn't. And here you have the number of cells, those ganglion cells per field. And here you're going to have the treatment. You have the PBS injection in the non-glaucomatous and glaucomatous eye. And you can see with glaucoma, the number of ganglion cells goes down. But when you injected the myanox cells, we did not see any loss of ganglion cells. And you can do also a measure of the thickness of the nerve fiber layer, and we also saw that thins when you induce glaucoma, but that thinning is prevented by the injection of myanox cells. All right, so we are at the point where if exogenously added myanox cells are neuroprotective in a model of light damage in albino rats and in a model of glaucoma, and these are neurons, what will happen in the brain? So for this, we use a model of, it's an interesting model of dementia and plaque formation that was developed in Australia by Jonathan Stone. And what they do is they use a needle that goes all the way through the neocortex and the hippocampus. And they see that in a few days, plaques start to form. If you wait long enough, it's going to lead to dementia. The hypothesis behind this model is that as we age, the capillaries in the brain lose the flexibility that they have when you're young, and the force of the pulse will lead to microhemorrhages. Those microhemorrhages are going to lead to some accumulation of some of the formation of the plaques. And this is a well-established model, has been done for many years, and proved through years. But for us, it was interesting because you cause this damage with a needle, and that allows us to attach that needle to a syringe and inject myanox cells in there, right? So we can inject the myanox cells right after we cause the injury. And that was nice for us. And these are them, the approach that we did. We did the treatment, and we injected the myanox cells and got rid of the myanox cells, and we killed the animals after 48 hours. And what we found is that the addition of myanox cells reduced the number of neurons dying or apoptotic cells within the brain caused by that injury. And we looked at that, and that was exciting. So you can see the myanox cells, you don't have as much tunnel. So that was really good. So we published that. So that was a model outside the eye. So far, what we know is the myanox cells migrate to stress within the retina. The depletion of the myanox cells leads to increased neuronal cell death, and the addition of exogenous myanox cells leads to neuroprotection. So that was wonderful. So you wonder, these cells are great. Why don't we use them in therapy? Let's take them out. So there's two problems. The first problem is that the myanox cells that we take them from a brain of another mouse or another rat, and you don't find many people volunteering for that. And the other problem is that cell therapy in general is complicated to get approved. But the biggest problem is that there's dark side to myanox cells that I'm going to talk to you about. So not everything is good. And I'm going to show you what the problem is with the myanox cells. So the first studies that we did that showed that we had to keep an eye on them is the studies on the lens. So we saw myanox cells in the lens. And you need to know that when people get cataract surgery, a percentage of the patients that get cataract surgery will develop what is called a secondary cataract. The technical name for it is posterior capsule opacification. So when you do cataract surgery, you open the capsule and you remove the tissue inside, and then you put an artificial lens in there, but sometimes the capsule opacifies. and that is caused by myofibroblasts that lead to wrinkle of the capsule. And we know that myanoid cells turn into myofibroblasts. So what we were looking at is at these myofibroblasts is the source of these myofibroblast myanoid cells, and what we found is that indeed they are they express BA1 or G8, which is the same. We just didn't know at the time, and that they are surrounded those wrinkles, and probably the reason, and they express contractile proteins such as smooth muscle actin. So they are in the wrinkles, and they are probably the source of the wrinkles. And so we said, okay, well, let's see if we can get rid of them again, of the myanoid cells. By then, we partnered with a company called Genisphere. We wanted to develop a more sophisticated approach to getting rid of the myanoid cells. So they design some dendrimers, DNA, 3 DNA structure molecules, and they can put an antibody against myanoid cells. This is the one over here, which is the G8 antibody, and they can put doxorubicin, which is going to get into the cells when this binds, the G8 binds to the myanoid cells. They're gonna get that structure inside the cytoplasm. The acidity is going to break the DNA, and the doxorubicin is going to be released, and that's going to lead to the killing of myanoid cells. We did the studies in vitro to prove that this worked, and it worked really well. So we used a rabbit model of posterior capsule opacification or secondary cataract, and you can see the wrinkles here forming in the lens, and you can see a lot of those wrinkles are caused by cells expressing smooth muscle actin that leads to contractility. And when we got rid of the myanoid cells, our 3-DNA dendrimer, as you can see here and here, you can see much fewer wrinkles being formed and fewer expression of smooth muscle actin. We can assess PCO, posterior capsule opacification, in these rabbits, and we do that blind, of course, and you can see that when we got rid of the myanoid cells, the level of PCO really was reduced. This is an example. On the left, you have PCO on a rabbit. You can see the opacification, mostly pearls. Pearls is what is called these white dots over here that lead to opacification. And these are two different concentrations of the 3-DNA dogs against myanoid cells. You can see here that removed 70 micrograms. We saw almost no opacification in the body. All right, so that means that the myanoid cells are bad. They can lead to opacification of myofibroblasts. Another pathology in which we were interested and we thought they might play a role because they can turn into myofibroblasts is PVR. So PVR stands for proliferative vitreoretinopathy. This is a complication that comes out of a retinal detachment, and a membrane forms in the vitreous side of the retina, and sometimes in both sides. And that membrane is usually formed by myofibroblasts. It's a contractile membrane and can lead to a secondary detachment and some retinal faults. And that is what is PVR. And you can see this image of the fundus of the retina, and you can see the membrane that has formed there and impedes the light going through. All right. So this was interesting for us because we thought, well, it's fibrosis. Myofibroblasts are forming that epiretinal membrane. It's quite likely that the myanoid cells are going to play a role there because we saw that they happen in the lens. The problem is that right now, the most popular hypothesis for PVR formation is that the cells that are right above, on top of the photoreceptors, are called the retinal pigment epithelial cells. When they break in the retina, those retinal pigment epithelial cells go into the vitreal side, because they're in the opposite side. They go into the vitreal side, and then they turn into myofibroblasts, and they form a scar tissue, and that tissue leads to a secondary detachment. The problem with that theory is that not all pre-retinal membranes form after a detachment. Sometimes you have them form without a detachment. The RPE cells have never been shown to express contractile proteins in vivo. That's what it should say, in vivo, as far as I know. They did some studies in vitro what they did. And also ultimately it's that all animal models for this are very invasive. It involves injecting human cells into RPE cells into a mouse or form a break. We thought myanox cells played an important role in this. So we wanted a model that didn't have this break, but still led to epiretinal membrane formation so that we can separate the aspect of the retinal epithelium from the myanox cells. All right. So we think that myanox cells are turning to myofibroblasts and driving the tractional forces that lead to the detachment and membrane formation. So can we develop a non-invasive model, marine model of PBR? And for this, you just have very bright medical students that do a lot of work. And what we found is that this protocol is that the animals, when they're born, they place a 65% oxygen chamber. And then 7 days after that, they place in normal oxygen, okay? And then we use OCT images. Remember the OCT is the machine that, you can live image the retina and measure the thickness at different time points to see how is it doing. And then at the end, after a couple of months, you kind of take the eyes and look on the microscope for myofibroblasts. So this is what we saw. So we saw that by week 4, 65% of the animals had developed epiretinal membrane and some kind of disruption in the inner part of the retina. In some cases, we have from this 65% oxygen, just 1 to 1% oxygen. So this is a control. This is the experimental. This is no percentages. I will show you the numbers later. In week 5, you have those epiretinal membranes formation. You can have some loops that are forming invaginations, kind of like horseshoes that are forming within the retina. And you can see a start of a retinal detachment here. And you can see by week 7, a lot of them have this retinal detachment that have formed. And this is just by using oxygen, right? We didn't cause any disruption. We didn't break the retina, nothing like that, right? So these are the numbers of the percentage of eyes that develop either epiretinal membrane or retinal detachment in white. So you can see that almost 80% of the animals develop epiretinal membranes, and that with time, those epiretinal membranes, because they have contractile proteins, they will start leading to small invaginations of the retina and retinal detachments in white. Okay. By week 7, almost 25% of the animals were showing that. This is what it looks like under the microscope. We label for myanox cells with an antibody called BA1, is what you can see here. Any cell in red is a myanox cell. And here you can see a smooth muscle actin expression for contractility. You have the epiretinal membrane form at the bottom. And you can see there's a lot of myanox cells leading to this loop in the retina. This is not normal. This should be a continuous, but you can see there's a shoehorn that eventually will lead to a retinal detachment over there. And that's because of the traction that the myanox cells express in a smooth muscle actin are forming there. And this is without the retinal epithelium. Retinal epithelium is up here. It hasn't gone over here. So this is independent from that. So I'll show you that the myanox cells are great. They're neuroprotective in some cases, but the problem is they can also lead to fibrosis and retinal detachments, right? So that is a problem, right? So for us, it was important to separate this and see if there's a factor that they express that can lead to neuroprotection. Our prime candidate for this was noggin. Noggin is binds to BMPs as an inhibitor of BMPs. BMPs are very important for the development of the embryo. And it's one of the key parts of the development of the eye as well. And it can induce neuronal tissue to regenerate. Some studies that we looked at were really interesting. So this study done Northwestern University was looking at the effect of noggin, injection of noggin in a model of stroke, permanent cerebral arterial occlusion. As you can see here is the effect of the stroke right here. And they injected noggin, and when they injected noggin, they saw that the volume of the infarct was much smaller. In addition to that, to reducing the infarct, what they saw is that there was animals with noggin did not show any motor deficit compared to controls, which showed a lot of motor deficit. So it was an interesting way to start. So we thought we used noggin as a good candidate for us because it's expressed for the minor cells, and see if we can find out what is that they're expressing that is leading to neuroprotection. Right? So for this, we use a model of retinitis pigmentosa. Is the one I told you about earlier. Is the RD1 mouse. This is a normal retina. In this case, it's inverted to what I showed you earlier. You can see the ONL here. You have the photoreceptors. You have the inner nuclear layer. This is the ganglion cell layer. And you can see how in these animals, the outer nuclear layer is almost gone as they become blind by 6 weeks. Okay, this layer over here, they just turn into that. So that's where the degeneration happens the most. So this is the approach that we had. So we treated the animals at 2.5 weeks, and we injected them with either noggin, PBS, or MSA, that's mouse serum albumin. That's a protein control for the noggin injection. We injected 5 eyes, 5 different ones, and we did functional studies, the ERGs, as you can see here, and then we killed animals at different time points, at different weeks. I'm not going to show you each one of those weeks, but basically it's the same result. What we found is that the retinas that were injected with noggin, they showed a significant reduction in retinal cell death when compared to controls. And you can see here is these 3 bars are week 3, and these 3 bars are week 4. These are the 3 treatments. So you have PBS treated in black, you have a mouse serum albumin here in red, and you have the noggin injected retinas in green. And there was a significant difference between total cell death in the retina when you injected noggin compared to the others. Where was that? So here we just looked at the distribution of those tunnel positive cells, those cells that were apoptotic within the 3 layers of the retina, the outer nuclear layer, the inner nuclear layer, and the ganglion cell layer. These are the 3 treatments, PBS, mouse serum albumin, and noggin. And you can see noggin in particular in the outer nuclear layer, you saw a reaction on the number of cells that died there. There was also a significant difference in the inner nuclear layer. When the photoreceptors died, some of the inner nuclear layer and ganglion cells are gonna die just because they're connected synaptically. And that happens at week 5, week 6, week 3, week 4, and we can look at the stress in these retinas by looking at GFAP. GFAP is a protein that is expressed by the glial cells, in this case the Mueller cells, in the retina mostly, and the expression levels relate to stress. And you can see there's a lot of stress in these retinas because the photoreceptor layer, which is up here, has almost disappeared. And the Mueller cells are panicking and expressing a lot of GFAP. And you can see the stress levels is much lower when you inject noggin. We can measure the thickness of the retina. As I told you earlier, that when animals get blind, the retina starts to become thinner and thinner. Again, the animals that were injected with noggin are the ones in green. The farther up this line is, the thicker the retina is. So you can see the other controls have thinner retinas, and that happened across week 3, week 5, week 6. So that was great. So noggin seemed like a good candidate. It seemed like the one that we wanted to go for. So what we are gonna do next is we develop a conditional knock-in Krelox through CRISPR system for noggin, in which we can conditionally remove noggin from the mice with a Krelox system. In our case, because it's a knock-in, we put a tag a green tag when noggin is expressed. But where recombination occurs and noggin is not there, then we put a red tag. So we're still working on these mice, and we're trying to do the crossing and get them to breed. But yeah, so far, when we looked in the microscope, we see the expression of green there. So that's what we're moving into, so we can see if the myanox cells from these mice do not have neuroprotective effect, and what we're looking at is just caused by noggin. All right, yeah, this is the work of many people. Jonathan Stone in Australia and Alice Brownlee and Siva were very helpful with the light damage studies that we saw, and some of brain studies. They hosted me in the lab when we did this. Glaucoma project was mostly driven by Paul Lecker over here. As you can see, Chris was the person that did a lot of studies on the oxygen-induced retinopathy model. And Carlos Font is a master student that was in my lab that did a lot of the studies that you have seen on noggin, and just submitted his thesis as first on the oven. So thank you. I'll be happy to take any questions you might have.
Hello, everyone. My name is Alex. I think we will skip questions for today's session because we're past 3. But for everyone that is here at the room or at Zoom, if you have any questions, just let us know, and we will make sure that those questions get to Dr. Arturo. Before closing the session, I just want to say thank you, Arturo, for taking the time to present and share your research with us. And just hope to see everyone on the next one. Thank you. Bye-bye.
Thank you.